Hello guys, so welcome back. Today we shall be discussing another very important topic for your exams, right? I hope all of you know that is regarding the meningitis, right? So the topic is Neisseria Meningitidis. So we shall be discussing the properties of Neisseria meningitis, the general properties, and most importantly how Neisseria meningitis causes meningitis for you. Not only meningitis, this Neisseria even causes the remaining diseases as well, remaining associated conditions as well. So we shall look into all of that, and finally we will end the topic by discussing the treatment. So first of all, let us discuss the general properties of this bacteria, right? So we are discussing the general properties of this bacteria. So what are the general properties of this bacteria, guys? So the first general property of this bacteria, first of all, what is this bacteria? This is Neisseria meningitidis. The other name for this is Meningococcus, right? So I can also call this as Meningococcus. Meningococcus. So the other name for this is Meningococcus. Right. So this particular Meningococcus, guys, point number one, this Meningococcus has got a thin peptidoglycan layer. So what do you mean by thin peptidoglycan layer? I have been telling you all the time that those bacteria which have got thick peptidoglycan layer, they are called as gram positive, and those bacteria which have got thin peptidoglycan layer, so which have got thin peptidoglycan layer, they are called as gram negative bacteria. So this Meningococcus is a gram negative bacteria. Second important thing, not only that, this Meningococcus, is it a rod shaped or a cocci or a round shaped? Obviously by the name you can see that Meningococcus it means it is a round shaped bacteria. If you look at the shape of the bacteria, this is how it looks. So this is the bacteria, right? So this is Meningococcus. Okay, so Meningococcus is a diplococcus bacteria, right? Because they are in two, so they are in a pair. This is called as diplococci. And you know exactly how do they represent? They represent coffee bean shape. Okay, they represent coffee bean shape. That is why we call this bacteria as diplococci, coffee bean shaped bacteria, right? So they occur in pairs basically. So this is Meningococcus. Next, third important thing which you need to understand is that this bacteria does not have any kind of flagella or any kind of cilia. So it is obviously, when it does not have any kind of flagella or cilia, do you think this bacteria can move? No. So this is a non-motile bacteria. Point number 4 is that there are some species in bacteriology that is called as clostridial species, right? So you have got Clostridium tetani, difficile, perfringens and all, right? So these Clostridium bacteria, they release spores. So that is why they are called as spore forming bacteria. But this Neisseria meningitidis or the Meningococcus simply, it does not release any kind of spores. So it is a non-spore forming bacteria. So this bacteria is a non-spore forming bacteria. Okay. Next, fifth important point is that this bacteria releases an enzyme called as catalase. So if it is releasing an enzyme called as catalase, this bacteria is said to be called as catalase positive. This is catalase positive bacteria. In the same way, this bacteria also releases another enzyme that is called cytochrome oxidase. So this bacteria is called as oxidase positive. So out of both of these positivities, it is very important to know about the catalase positivity, right? So for example, let us say this was your bacteria, right? This was your bacteria. This is your diplococci. Now this bacteria is releasing a special enzyme. Okay, and that enzyme is called as catalase. That enzyme is called as catalase. Can anyone tell me what this catalase will do? Yes, this catalase will convert H2O2. H2O2 is hydrogen peroxide. So it converts H2O2 into water plus half mole of oxygen. It is converting H2O2 into water and half mole of oxygen, right? So hydrogen peroxide is breaking down into foam. So that is the reason why whenever you add hydrogen peroxide to your superficial wounds, you find foam coming out of that. Foam is nothing but it is having air bubbles, which is oxygen, and the remaining is the water. But always remember, never ever add hydrogen peroxide to deep cut wounds. Why? For example, let us say this is a blood vessel like this, right? This is a blood vessel. Now these are the endothelial cells. These are the endothelial cells. Let us say that some of the endothelial cells are damaged like this. The blood vessel got rupture. Let us say this is a deep artery. For deep arteries means femoral artery, brachial artery, radial artery, right? Popliteal artery. These are deep arteries. When you have very severe injuries, there might be a cut of these deep arteries. So when these deep arteries are cut, what is happening is that from the cut edges, catalase enzyme is released. Okay, now you are adding hydrogen peroxide. When you add hydrogen peroxide will dissociate into water and half mole of oxygen, isn't it? So within the blood vessel you will find many oxygen molecules like this. So all the oxygen molecules join together to form a very big air bubble, and that what causes air embolism in a patient. So that is the reason why never ever add hydrogen peroxide to deep cut wounds, because if you are adding hydrogen peroxide to deep cut wounds, there is a high chance that the patient might end up having air embolism. Air embolism. So never ever add hydrogen peroxide to deep cut wounds. Always add hydrogen peroxide to superficial wounds. So it is catalase positive, it is oxidase positive, it is moreover diplococcal. And next important thing which you need to keep in mind is that this bacteria lives only and only in the presence of oxygen. If there is some environment where there is no oxygen available, then this bacteria is going to die. So this is an aerobic bacteria. So it needs oxygen. Definitely it needs oxygen. So such aerobic bacteria is called as obligatory aerobe. This is an obligatory aerobe, right? So this is called as an obligatory aerobe. So what are the 7 properties, guys? It has got a thick peptidoglycan layer, right? Thick peptidoglycan layer, so gram positive. It is non-motile, it is non-spore forming, and these are very important, guys. Catalase positive and oxidase positive. Why are you so stressing on these two? I will tell you later on why it is so important. And it is an obligatory aerobe. All of you know. So what shape is this, guys? Obviously it is a coffee bean shape. Right. So now if I want to culture this bacteria, how am I going to culture this bacteria? If I wanted to culture this particular bacteria, then how am I going to culture this bacteria? How am I going to culture it? I will culture this bacteria on a specific medium, guys, right? I am going to culture this bacteria on a specific medium, and that is called as Thayer-Martin medium. Thayer-Martin medium. This medium is called as Thayer-Martin medium. The other name for this Thayer-Martin medium is also called as VPN medium. Can you please tell me what is this VPN medium? Why are you calling it as VPN medium? Look here, all of you know that this bacteria will grow well on Thayer-Martin medium, and I am telling you, on Thayer-Martin medium there is only one bacteria which grows, that is Neisseria meningitidis, right? This meningococcus. So this is the only bacteria which grows on Thayer-Martin medium. Now you might get a doubt, why not other bacteria? Obviously it is a growth medium, it is a culture media. Why don't other bacteria grow? Yes, other bacteria also can grow, but you are suppressing that bacteria by adding VPN. What is VPN? V stands for vancomycin, P stands for polymyxin, and N stands for nystatin. So you use these three antibiotics to inhibit the growth of what? The growth of the bacteria within this Thayer-Martin medium. Okay. So all of this, what they do is they inhibit growth of other bacteria. Where? In this medium. In this medium. So culturing where you do in the Thayer-Martin media. That is one thing you need to know. Another important thing is that this bacteria, right, we basically tell that this Meningococcus bacteria it ferments maltose, right, because in the previous lectures which I have spoke, you have seen that some bacteria they ferment lactose. In the same way, this bacteria can ferment maltose. So another important property of this Meningococcus is that, look here very carefully, this Meningococcus bacteria, what is it going to do? Look here now. This Meningococcus, what does it do is, this Meningococcus releases an enzyme, and that enzyme is called as maltase. Okay, and what is this maltase going to do? Anyone have any kind of idea? Yes, what this maltase is going to do? Maltase is converting maltose all the way to glucose. Okay, and what is happening to glucose? All of you know very well that glucose will take part in glycolysis, right? So in glycolysis you have got glucose is converted to glucose 6-phosphate, then fructose 6-phosphate, fructose 1,6-bisphosphate, right, 3-phosphoglycerate, 2-phosphoglycerate, phosphoenol pyruvate. So there is a big cycle which you will study in biochemistry anyways. So glucose is finally, after all this entire cycle, it is getting converted into lactic acid. Okay, so glucose, after a number of reactions, it is getting converted into a special thing that is called as lactic acid. This is an important thing. So finally, can I tell that from maltose to glucose, this is called maltose fermentation. From glucose to lactic acid, this is called glucose fermentation, right? So can I tell Meningococcus has a ability to ferment both maltose and glucose? Yes. And if I ask you, what is the end product of its fermentation? You can tell the end product of maltose fermentation is glucose, the end product of glucose fermentation is lactic acid. But anyways, we call this entire step till the lactic acid as maltose fermentation. So if someone asks you that, look, Meningococcus performs maltose fermentation. So can you tell me what is the end product? The end product is always lactic acid. Okay. So Meningococcus leads to lactic acid formation. Right. Now, on the other hand, there is another bacteria called as Gonococcus. And what does this Gonococcus do? Yes, Gonococcus ferments only glucose. What does Gonococcus do, guys? It can ferment only glucose. This can ferment only glucose, not more than that. Okay, Gonococcus can ferment only glucose, not maltose. So these things you need to keep in mind. So if you want me to write it, this is very important for your MCQs, guys. Meningococcus, and on the other hand we have got Gonococcus. Okay, so what does Meningococcus ferment? Yes, tell me. There are two things. One is called maltose, another one is called as glucose. Now you tell me, will Meningococcus ferment maltose? it will ferment maltose. It will also ferment glucose. But gonococcus will not ferment maltose, it will only ferment glucose. So this table, this thing you have to know it guys at any cost. Okay, fine. Now another important thing is that you told me that meningococcus ferments maltose into lactic acid, right? How am I going to know it? I want to look at visually that meningococcus is releasing an enzyme called maltase, and this maltase will convert maltose into pyruvic acid. I just want to know it. How am I going to know it? How am I going to look at it? How am I going to look at it is, first of all, you have to take a test tube like this. Okay. So in this test tube, you add two reagents. One is called as phenol red. So that is the reason why I am drawing it in red in color. Okay. So you add phenol red. Along with the phenol red, you also have to add maltose. Okay. So what did I add in this? What did I add in this test tube guys? I have added phenol red. I have added phenol red plus maltose. Phenol red and maltose. Now look what I am going to do. Now I am going to add this bacteria. What bacteria? I am going to add Meningococcemia into this. Okay. I am going to add Meningococcemia. And this Meningococcemia, once it enters into that medium, that phenol red and maltose medium, what it is going to do is that this Meningococcemia will release an enzyme called maltase, and this maltase will convert maltose into lactic acid. It means within the test tube, the acid content is increasing, the pH is decreasing, the acidity is increasing. So when the acidity is increasing, the red color will turn into yellow color. So now the test tube from the red, it will turn to another color. Look, what is that? This is a test tube. Now the test tube will turn to completely yellow in color. Okay. So this is an experiment which you have to know. So this test is also called as maltase fermentation test. What is this test called as guys? This test should be called as maltose fermentation test. Now this test, I am telling you, it is very important for your MBBS exams. So they gonna ask you in your Viva maybe. So this will be very, very important. So maltose fermentation test, what is the resultant? The resultant is the entire tube, the things which you have taken like phenol red and maltose, they will be turning it to yellow in color. So this is the important thing which you need to note it down, right? So how am I going to get infected with this bacteria, with this meningococcus? What is the way of transmission? What is this transmission route? How am I going to get infected? By respiratory and oral secretions. By what secretions? By respiratory and oral secretions. Clear? Fine. Right guys, so we are discussing about the pathogenesis now. Now in this pathogenesis, what is the important thing? Let me form some small basics. Now sit back and relax, don't make any notes. So I just want to form some small basics, and later on I'll join all of them and I'll be teaching you the pathogenesis. Now what are these basics? For example, I have taken a bacteria like this, right? I have taken a bacteria like this, right? So this part which I have taken here, look, there are dots in between. This part which I have taken here, this is called as inner membrane of the bacteria. This is called inner membrane of the bacteria. Now adding upon this inner membrane, I have got some very important structures in this way. Now what are these structures? Don't tell me it is DNA, but what are these structures? This is called as peptidoglycan. This is called as peptidoglycan. Fine. Upon this peptidoglycan layer, I have got another membrane that is called outer membrane. Now how does the outer membrane look? Like this. So in this way the outer membrane looks. You see, I cannot draw all of that, but just think this entire thing till here. I mean, obviously it is surrounding the entire cell wall. So this is an outer membrane. Now on the surface of the outer membrane, look, there are some very interesting things in this way, like this. And there are also one more interesting thing in this way. You see, on the outer membrane, this entire thing here is called as outer membrane of the bacteria. Outer membrane of the bacteria. So for the bacteria, we have got an inner membrane. Surrounding that we have got a peptidoglycan. Surrounding that we have got an outer membrane. Outer membrane is a phospholipid bilayer. Now within this phospholipid bilayer, right, we have got some very round projections like this, red color projections, and next we have got an arrow mark projection. Now look here very carefully that this red color projection which I have drawn here, this is called as pili. And next, this arrow shape projection which I have drawn here, this is called as OP proteins, opacity proteins, right? So OP proteins. What are OP proteins means? OP proteins means opacity proteins. How many opacity proteins are there? There are two opacity proteins. One is called as OPA, another one is called as OPC. Opacity protein A, opacity protein C. I have drawn only one OP protein because both of them, the OPA, OPC, they perform the same function. So that is why I have drawn only one OP protein, showing that this is both OPA and OPC. Okay. So overall we are having inner membrane, we are having peptidoglycan layer, surrounding that we have got outer membrane, and within the outer membrane we have got pili and we have got OP proteins. Now out of all these, what is the important thing to remember? The important thing to remember is pili. The next important thing to remember is OP proteins. These are the two important things. Why? Why are they two important things? Because look here now, look here. For example, let us say that this picture which I am drawing right now here, guys, this is the host cell. This is a host cell. So this is your host cell. Now on the surface of the host cell, you have got very important structures. So this structure which is located on the surface, this is called as a receptor. Whatever receptor it can be, that doesn't matter. So this is the receptors that are located. There are two types of receptors. One receptors are like this Y shaped and other receptors are in this way. This is the shape of the other receptor. M shaped, right? Now look what happens. This was your bacteria. This was your bacteria, right? Diplococci. Now this bacteria is having pili. Now these pili fit into these receptors. Now the way this pili got attached into the receptors, this attachment is called as temporary attachment, or also called as loose attachment, or also called as initial attachment. So if I ask you, initial attachment is done by initial attachment on the host cell, it is done by what? It is done by pili. This is an important question. Next important thing is that obviously they are also having this structure that is coming out anyways. They are also having this structure, that is arrow shaped structure that is coming out. But anyways, look here. In the next picture, what is happening is, this is your diplococci, and now this is the pili that are getting attached to the receptors. And next, look now what is happening. Not only pili, even OPC, that is opacity proteins. OP proteins, that is opacity proteins A and opacity protein C, even they are getting attached. Now the moment this opacity protein attaches to that blue color receptor which you can see, now this type of attachment is called as tight attachment. So if I ask you, tight attachments are done by, tight attachment is basically done by what type of proteins? Obviously OP proteins, that are opacity proteins. And just to remind you, I already told you how many different types of opacity proteins we have. We have got opacity protein A, we have got opacity protein C. Okay, so they get attached. Now the moment pili get attached, nothing is going to happen. It is very loose attachment. Next, after pili, the center part, if it is getting inserted there, that is the OPC, once it gets inserted there, this is called as tight attachment or the final attachment. Now the host cell will take up that entire bacteria into the body, into the cell inside, right? So this entire bacteria, it will fold inside like this. The bacteria was here, now the entire cell will take up that bacteria and throw it inside its body. Clear? Now how is it throwing inside and what is it happening? But before that, I'm telling you guys again and again, this is a very, very important thing. This thing which I have written, right? So this is a very, very important thing. You have to know it at any cost. Clear? Now basically how this is transmitted, I told you, through nasal or oral droplets it is getting transmitted to you. So if it is getting through nasal droplets or oral droplets, first the bacteria enters into the nasopharynx or the bacteria will enter into your oropharynx. So let me explain you the picture, let me draw the picture and then explain you in detail what is happening. Now concentrate on the pathogenesis part. Okay. So now let us say, for example, this is your lateral side of the face. This is your lateral part of the face. This is your lateral side of the face like this, right? So these are your eyes over here, and all of you know very well that this part which I am drawing right now, this part here, this is called as your nasopharynx. Nasopharynx. Okay. The next one, this part which I am drawing right now, this is called as your what? It is called as your oropharynx. One is called as your nasopharynx, next is called as your oropharynx. Your nasopharynx and oropharynx together they join and form laryngopharynx. Now within this nasopharynx, guys, you have got some small cells. These are called as epithelial cells, mucosal epithelial cells, right? So even in the oropharynx region also you have got these cells. Now what I'm going to do is that through the nasal droplets or through the oral droplets, this bacteria is entering into the nasal cavity, that is into the nasopharynx, or some of the bacteria is entering into the oropharynx. Now what I'm going to do is that I'm going to enlarge this one epithelial cell and look what is happening there in a bigger picture. Okay, so I'm just going to enlarge this out. I'm just going to enlarge this picture out like this, in this way. Okay, now look, this is your wall of your pharynx, right? Let us say it might be nasopharynx or oropharynx, that doesn't matter, but it is a wall of your pharynx. Now there you have got a bigger cell. I'm just enlarging one cell, right? So you have got a bigger cell like this. Let us say this is your cell. This is your cell. So let me darken it up. Let me darken it up so that it would be better for you to understand. So this is the base. This is the base, and onto this base there is attachment of what? There is attachment of this particular cell. So this is your mucosal cell like this, right? So this is your epithelial cell. Where this epithelial cell is located, guys? This epithelial cell is located on your nasopharynx or oropharynx. Now what will happen? I already told you that you have got some filaments like this, I mean some receptors in this way. So with the help of these receptors, there are two types of attachment I told you. One is called as initial attachment, another one is called as tight attachment. So let me draw it up first. I'm not going to explain you now again. You know it, right? So now both of them have got an arrow shaped structure like this. This is called as your OP protein, right? Now from here comes one of your pili in this way, and from here comes your another pili. Same way on the other side, pili on this side, pili on that side. Next, here I should have some receiver, someone to receive this, right? So the next picture with the golden receptor which I am drawing, this is called as tight attachment, whereas this one is called as initial or loose attachment. Now, will the mucosal cell, will it accept the tight attachment or loose attachment? Obviously, it will accept the tight attachment. So in this way, the bacteria enters inside your mucosal cell. Now your bacteria is within your cell. Now the moment bacteria enters into your cell, all of you guys, you have studied this well in your MBBS first and second years. All of you know that in your mucosal secretions, either it can be the nose or the nasopharynx or the oropharynx, whatever it is, in your mucosal secretions, what is that barrier which you have? What is that antibody barrier which you have? Yes, it is IgA. Very good. So here, immediately as the bacteria enters inside, the IgA molecules, they start coming like this. They start coming like this, and they think that I have to attack this new bacteria which is entering into my body. Now, looking at the IgA, this diplococci or this meningococcus, I am calling it as meningococcus from now. So looking at this IgA, this meningococcus is so afraid that this meningococcus immediately starts releasing some very important enzyme. Look here. This meningococcus starts releasing a very important enzyme that is called IgA protease. That is called as IgA protease. What is that enzyme name called as? That is called IgA protease. You know what does IgA protease do? Whatever IgA is coming to, for example, if I am the bacteria, whatever IgA is coming and trying to kill me, I will release IgA protease and break down that IgA. So, as per the concept what I told you, what is happening right now? All these IgA molecules are broken down. So this IgA molecule is broken down, this is broken down, this is broken down. But I left one IgA molecule, it is not broken down. What I am trying to explain you? What I am trying to explain you is that if 100 IgA molecules are coming, just only 4 or 5 IgA proteases are released. And these 4 or 5 IgA proteases will kill down, let us say 50 or 60 IgA molecules. But the remaining 40 IgA molecules are left like that, left undestroyed, they are not destroyed. So what will happen now? What will happen? What will these IgA molecules which are not destroyed, what are they going to do? What are they going to do is that, listen very carefully, these IgA molecules, they simply put a tag on this bacteria. What are you talking? What do you mean by tag? So they put a tag. For example, there is a dark room. I am in the dark room right now. And exactly right side to me, there is a thief. Now, near to the window over there, there is a police. Now the police don't know who is the thief. He can't even see him. So what I do is, I put a radium tag on his t-shirt. When I put a radium tag on his t-shirt, then the police can easily identify him and can catch him and can arrest him. So the same way IgA molecule is doing. IgA molecule will put a radium tag on this diplococci or meningococci. The moment it puts there, the neutrophils in your body can very easily look at that diplococci and they will rush to the diplococci and start eating up the diplococci. Clear? Right. Now you can ask me, what is this process you are explaining? The process where IgA is making these meningococci visible to your neutrophils, this process is called as opsonization. What is opsonization? What is opsonization? This IgA performs a process called as opsonization. Opsonization is, it is a process by which it is showing those meningococci, it is exposing those meningococci to the neutrophils so that neutrophils will come and attack them fastly. So in this process of opsonization, IgA should put a tag, it should put a radium tag. And what is that radium tag? Those radium tags are called as opsonins. Those radium tags are called as opsonins. Those are called as opsonins. Can you tell me what are those opsonins? For example, opsonins like IgG. IgG is an opsonin. Not only IgG, there is also one more thing that is called as C3b. C3b is also an opsonin. The next one is mannose binding protein. The next one is mannose binding protein. So we have got three types of opsonins. One is IgG, C3b and mannose binding protein. If you ask me, if you get a doubt that are only these three opsonins, no, there are many more opsonins, but these are the three major opsonins in your body. These are the three major radium tags in your body, right? So finally, out of these three, this IgA have added one tag. Let us say the IgA, I think it has added a tag called IgG onto the surface of the bacteria. Now the moment it added that radium tag onto the bacteria, the neutrophils start rushing there now. Okay, the neutrophils start rushing there. Now all the neutrophils will come here. Now look here. Let me draw one neutrophil. Let us say this is a neutrophil. Okay. So right, this is the nucleus of the neutrophil. Let us say you got a neutrophil here. Now what this neutrophil will do, guys? This neutrophil will engulf that bacteria, isn't it? So this is a diplococci over here, and diplococci or meningococci, it is the same. Don't confuse. Sometimes I am using meningococci, sometimes I am using diplococci. Both of them are same. So there is diplococci over here, there is a meningococci. So this neutrophil will engulf. Okay, let me write it, guys. Let me write this thing that it is a neutrophil. Now this neutrophil will engulf that. Now when it will engulf that, how will the picture look? This is going to engulf that in this way. In this way. And finally, we have got a phagosome like this. Okay. So let me enlarge this phagosome, guys. Let me enlarge this phagosome. We have got a phagosome. Now this thing which I am drawing here, guys, don't confuse, this is a neutrophil within a neutrophil. No, this is a phagosome. Why the phagosome is so big? I have drawn it like that to explain some things in that, right? So finally, your diplococci here are in the phagosome now. Okay, they are right now in your phagosome. Now you know what happens here? What happens here is that, guys, what is the important thing you need to know is that this phagosome, within this phagosome, there is H2O2, that is hydrogen peroxide, right? So guys, let us brush up all these things. Bacteria in the, very fastly, bacteria in the nasopharynx. From the nasopharynx, with the help of pili and with the help of opacity proteins, it got attached to the surface. And now the cell has taken up inside. Once after taking up inside, all the IgA came there. Now this bacteria started releasing bullets, that is IgA protease, and killed, let us say, 60 or 70 IgA molecules. The remaining 30 IgA molecules, they put a tag on this bacteria so that neutrophils can identify them and neutrophils should come there. So obviously neutrophils came there now. After coming, these neutrophils have engulfed this bacteria, phagocytosed this bacteria. After phagocytosis, the bacteria is stored and kept in a vesicle called as phagosome. So this picture here, this is your phagosome. So finally the bacteria is in the phagosome. Now within the phagosome, we have H2O2, that is hydrogen peroxide. What will happen is that hydrogen peroxide is converted into OH dot. What do you mean by OH dot? OH dot means hydroxyl radical. All of you look here now. H2O2 is getting converted into OH dot. This is called as hydroxyl radical, or simply called as reactive oxygen species. Simply called as reactive oxygen species. And these hydroxyl radicals or superoxide radicals or whatever radicals are there, these radicals will kill down the bacteria very fastly. They will cause lysis of the bacterial cell membrane. So within this phagosome, what is the plan of this phagosome neutrophil is that I will take the bacteria inside and I have H2O2, I will convert it into hydroxyl radical and this hydroxyl radical will kill the bacteria. But bacteria is so clever. You know why? I told you in the starting that bacteria is catalase positive. It means bacteria releases an enzyme called as catalase. So when it releases catalase, all of you know what is going to happen. This catalase will convert your H2O2 into water plus half mole of oxygen. Now, will this water and oxygen kill the bacteria? No. So now the bacteria thinks I'm safe. Now the bacteria starts to divide. They divide. Finally, look what is going to happen. This bacteria divides in this way. Even the bacteria comes out of the phagosome and even it starts dividing here. So in this way, the bacteria divides like this. So now the entire neutrophil is with the bacteria. Now the neutrophil has lost its chance. Now the entire neutrophil's power is gained by these diplococci. Now diplococci are ruling the neutrophil now. So the entire neutrophil is taken up by these diplococci, and diplococci they divide and finally neutrophil has got no space, then neutrophil will burst out. Okay. What is going to happen when neutrophil will burst out? Look here now. This is your basal surface of the cell. This is your basal surface of the cell, right? And let us say that this was your cell surface like this. This was your cell surface. Okay. And obviously guys, if you look at this picture, here, let me rub these two golden lines. Now back of the pharynx, you have got blood vessels, isn't it? See, there is a blood vessel like this. Obviously, this blood vessel supplies blood to the nasopharynx and oropharynx. So in the same way, can I draw a blood vessel here? In this way? Yes, I can draw a blood vessel like this. Back of the pharynx, right? Now look what is happening. So this is your neutrophil. The neutrophil is completely loaded with the bacteria and the bacteria still divides and finally the neutrophil bursts out. Look now, the neutrophil bursted out. Now when the neutrophil bursted out, whatever diplococci are located inside this neutrophil, these diplococci are very freely released into the cell. Not only into the cell guys, these diplococci are so many in number that diplococci will very easily enter into the bloodstream like this. Right now diplococci are in your bloodstream. Okay, look at this smaller picture. Now the diplococci are in your bloodstream like this. Now having the diplococci in the bloodstream leads to a condition, or this condition where the diplococci is seen in the bloodstream, this condition is called as meningococcemia. Meningococcemia. Clear all of you? Now look, one more thing. Bacteria is exactly here. So you have got a brain here. Obviously all of you have got your own brains, right? What if this bacteria will travel a very short distance and enter into the brain? If this bacteria from the blood circulation of the pharynx, that is the nasopharynx and the oropharynx, which you can look at the picture here, from here if the bacteria goes up, it ascends up into the brain, what will happen? It will cause a condition called meningitis. It will cause meningitis. Obviously all of you know that this thing is going to cause meningitis. Keep this thing in mind. So far we have learned two diseases. One is called meningitis. I'll tell you what are the MCQs that are asked here guys. So first listen to the concept basically. One, it is called meningitis. It goes up, causes meningitis. It goes back, enters into the bloodstream and it causes meningococcemia. And not only that, what all things it can cause? What all things it can cause is that, look here very carefully. I'm just enlarging the blood vessel now. This is your blood vessel, right? So this is your blood vessel over here. All of you know, there is no surprise here that your blood vessel is having endothelial cells. I'm telling you again guys, learning the concepts in much detail is very important for you, because you are not appearing now for your NEET-PG exams or next exams. You have a lot of time. But before that, if you learn all these concepts now during your MBBS, it will be really very helpful for you. Not only I am covering the microbiology, so through these concepts you can even cover the remaining subjects. When you go to the remaining subjects, everything will be very easy for you. So it is better to spend more time in learning the concepts. Right, so these are your endothelial cells, all of you know, right? So simple thing guys, I have not done anything. I have just enlarged the blood vessel to show what is this bacteria doing in the blood vessel, that's it. So this bacteria guys, now this bacteria is here, right? Now what this bacteria does, that is an important question. Now this bacteria releases toxins. And what are those toxins guys? This bacteria releases endotoxins. So this bacteria releases toxins. What toxins does this bacteria release guys? This bacteria releases endotoxins. Now, what can be the question in the exam regarding endotoxin? Apart from the functions of endotoxin, I'll explain you very clearly the functions of endotoxin. But in the exam, they'll ask you from where endotoxin is released. You are telling bacteria is releasing endotoxin, but from where is it released? Where is it released is that, for example, if this is your bacteria, all of you know, I have already drawn this on the surface of the bacteria. What is this layer guys? Tell me now. Very good. This is your peptidoglycan layer, right? So one becomes the inner membrane, next peptidoglycan layer, and after that you have got another layer like this. What is that layer called? Outer membrane. Within that outer membrane, I told you there are pili and also there are OP proteins, right? Opacity proteins. Not only that, if you zoom into this outer membrane, only outer membrane, you will find three important things. What are those three important things? You will have a small string like thing like this. After that, you will have a slab. Then you will have small rings. In this way. What is that string like thing which I have drawn guys with in green in color? That is called as O antigen. O antigen. What is that slab which I have drawn? This is called as core polysaccharide. This is called as core polysaccharide. And the final one, the golden one, this is called as lipid A. This is called as lipid A, and lipid A is nothing but your endotoxin. Lipid A is nothing but your endotoxin. Okay. So if someone asks you, right, you are a student, if someone asks you, can you tell me from where in Meningococcus, from where endotoxin is released? Not only in Meningococcus guys, any gram-negative bacteria, if it is releasing endotoxin, then if I ask you from where endotoxin is released in gram-negative bacteria, the answer will be bacteria has got an inner membrane and outer membrane. Outer membrane has got three parts. One is called as O antigen, core protein, that is core polysaccharide, and lipid A. Lipid A is nothing but called as endotoxin. It means this entire unit like inner membrane, outer membrane, that peptidoglycan, everything together you call it as a cell wall. So when I am breaking the cell wall, the endotoxin is released out. Endotoxin is not coming from within the bacteria, it is coming from outside the bacteria, from the cell wall of the bacteria, right? From the cell wall of the bacteria. So that is what is called as your endotoxin. This concept which I have explained to you about O antigen, core polysaccharide, and lipid A, this applies to all gram-negative bacteria except Neisseria meningitidis. Why? This entire three layers, this outer membrane consisting of three layers, right? These three layers together guys, all these three layers together, you call it as lipopolysaccharide. lipopolysaccharide. Okay. So this lipopolysaccharide is present in all gram-negative bacteria except this Neisseria meningitidis. Why? Because in Neisseria meningitidis, you look at the inner membrane, you look at the peptidoglycan, you go to the outer membrane, and in the outer membrane you won't have O antigen. There is core polysaccharide, there is lipid A, but O antigen is absent for Neisseria meningitidis. So when O antigen is absent, I am not calling it as lipopolysaccharide, but what should I call it as? I should call it as lipo-oligosaccharide. Lipo-oligosaccharide. Okay. So from this lipo-oligosaccharide, within this lipo-oligosaccharide, I have two components. One is called core polysaccharide, another one is lipid A. From this lipid A, my endotoxin is released. Okay, clear guys. This Neisseria meningitidis is having lipo-oligosaccharide. Okay, this lipo-oligosaccharide is having lipid A and it is having core P. Core P means core polysaccharide. And this lipid A is the one which releases endotoxin. Clear now? Right. What this endotoxin is going to do, guys, that this endotoxin, once this bacteria enters into the blood vessel, right, this bacteria releases that endotoxin. How should I draw that endotoxin? I'll draw it with dark green. So it releases these endotoxins from oligosaccharide of lipid A, right, lipo-oligosaccharide of lipid A. Now these endotoxins are phagocytosed by macrophages, obviously. So your macrophages are waiting here, and these macrophages, you see the macrophages are taking this particle inside. Now once this macrophage takes this endotoxin inside, this macrophage starts releasing two important compounds, two important chemicals, right, rather to say it is chemicals. So one important chemical is that it releases nitric oxide. The other important thing is that it releases interleukin 1, it releases interleukin 6, it also releases TNF alpha. What are the two things, guys, it is releasing? Yes, what are the two things it is releasing? It is releasing interleukin 1, interleukin 6 and TNF alpha. On the other hand, it is releasing nitric oxide. And all of you should know one thing, that nitric oxide will cause vasodilation. Nitric oxide will cause vasodilation. And what do this interleukin 1, interleukin 6 and TNF alpha do? What do they do is that, can you look at these endothelial cells which I have drawn here, guys? What I am doing is I am rubbing one endothelial cell. Okay, so I'll take away one endothelial cell. Now after taking this one endothelial cell, I'll just enlarge this endothelial cell here. Let us say this is one endothelial cell. Within the endothelial cell, I have got an important compound that is called as arachidonic acid. Arachidonic acid. And this arachidonic acid is converted to prostaglandin. Arachidonic acid is converted into prostaglandin. With the help of what enzyme? With the help of COX, that is cyclooxygenase. So now this interleukin 1, interleukin 6 and TNF alpha, what are they going to do? They directly go and activate your COX enzyme so much. So when they stimulate your COX enzyme, the levels of prostaglandin are increased. So what is the function of prostaglandin? Prostaglandin performs two functions. One is called as hypotension, another one is called as fever. Okay, prostaglandin performs two functions. So let me write all these things here, guys. Let me write all these things here. What is the thing? The thing is that we have got endotoxin. endotoxin enters into what? It enters into macrophage, and macrophage gets activated and it releases cytokines, right? So first of all it releases nitric oxide, then it releases interleukin 1, interleukin 6 and TNF alpha. Okay, so what is this interleukin 1, 6 and TNF alpha going to do, guys? Yes, right, tell me. So this nitric oxide, first of all it will cause vasodilation. So whenever there is vasodilation, whenever the vessel dilates, the blood pressure inside the vessel will fall down, and that is called as hypotension. Second important thing is that it will even activate interleukin 1, interleukin 6 and TNF alpha. So all these three are causing increase in level of what? Increase in level of prostaglandins. And what do these prostaglandins do? Prostaglandins do two functions. It does two functions. One is fever, another one is vasodilation. So when there is vasodilation, again there will be hypotension. So overall, what is happening with this guy? As a result of endotoxin release, there is hypotension, there is fever. Again there is hypotension. So patient is having hypotension and fever, right? So the more endotoxin release, the more hypotension and the more fever the patient will have. So for example, right now within 2 seconds there is heavy influx of endotoxins. Then the hypotension will be so severe that the patient's blood pressure will fall down suddenly, and that is called a shock. Okay, right. Next important thing, these endotoxins not only do this thing, but these endotoxins also does another thing. Look here. Second important thing, for example, this is your liver. Okay, now within your liver, let us say these are the endotoxins, right? So what were the endotoxins here? These were the endotoxins, right? So what did these endotoxins do is that these endotoxins started activating the liver. When they started activating, now don't think these are molecules of our body, these are endotoxins. Now these endotoxins, once they are activating the liver, liver releases two important complement proteins. What are those two important complement proteins? It releases C5a, right? Not only C5a, it also releases C3a. These are the two complement proteins. What are these two? These are the two complement proteins. So what these two complement proteins will do? Look here. These complement proteins will go all the way like this, and here you have got your cell, and this cell is called as mast cell. And what is this cell, guys? There are granules within the mast cell. So this cell is called as your mast cell. So what is happening in the mast cell? What is happening is, look now, this C5a and this C3a together, they have a very long injection like thing. This injection like thing will break down the mast cell. When it will break down or it will make holes within the mast cell. When it will make multiple holes within the mast cell, what is happening? What is happening is that all this histamine, whatever is located within the mast cell contains histamine, right? All the histamine comes out in this way. Can anyone tell what is the function of histamine? Histamine does vasodilation. So again, even in the second process also, there is vasodilation. Whenever there is vasodilation, patient will again have, yes, what is that the patient has? He has hypotension. See how many factors are contributing for hypotension. What if the patient is having heavy influx of endotoxins? By many mechanisms, the hypotension will go so severely, isn't it? The last mechanism that endotoxins are going to do, or the endotoxins are going to affect your body, are, look here now, the last mechanism. The last mechanism is that we have in our blood vessels, we have got something called tissue factor. Okay, we have got something called tissue factor. For example, This is my blood vessel, right? So within the blood vessel, I have got my endothelial cells like this. Okay. For example, let us say one endothelial cell is damaged like this. Okay, one endothelial cell is damaged. Let us say these endotoxins are causing damage of few endothelial cells. So when few endothelial cells are damaged, from that broken endothelial cells, tissue factor is released. What factor is released, guys? Tissue factor is released. And what this tissue factor does within the blood vessel, tell me? This tissue factor will convert factor 7 to factor 7a. That is activated factor 7a. And this factor 7a will convert factor 10 into activated factor 10a. And this activated factor 10a will convert PT, which means prothrombin, to a molecule called thrombin. All of you know this mechanism, right? So what thrombin will do finally? Thrombin will convert, yes, fibrinogen to fibrin. Now I don't want to write here, guys, because all of you know what does fibrin do. Fibrin will form platelet plug, right? It will call all the platelets and form a plug. Clear? Fibrin will cause blood clotting in your body. Simple terms to tell, fibrin will cause blood clotting in your body. It will call all the platelets, and all the platelets together, this fibrin will tie a rope around that, so that all the platelets will form together, forming a clot. In the blood, all the platelets are decreased now because all the platelets came here. So in the blood there are no platelets. So the level of platelets decreased. You're clear? You're clear here? See here. In this room there are 100 members. So all these people, if they go to the other room, the number of people in this room are 0. The same thing, in the blood, platelets are equally distributed. So if all the platelets in the blood, right, all the platelets are present in the blood, they are equally distributed. And because of this increased amount of fibrin, all the platelets are coming together to form a platelet plug. So in the blood over there are no platelets found. So patient will have thrombocytopenia, isn't it? Patient is having thrombocytopenia. Right. Patient will have thrombocytopenia. Apart from thrombocytopenia, what is the other thing? What is the other thing is that these endotoxins, these are the endotoxins, right? These endotoxins cause damage to the blood vessel, causing releasing of tissue factor, and these endotoxins will stimulate the tissue factor too much. They will activate the tissue factor. They will activate the tissue factor too much that there will be excess amount of fibrin production. When there is excess amount of fibrin production, the clots which are formed in your body are also excess, and that is called as disseminated intravascular coagulation, simply called DIC syndrome. Disseminated intravascular coagulation, which is also called as DIC syndrome. Clear? Right? So let me, guys, write down what all the things that this bacteria basically causes to you. The first thing which we discussed, guys, was from the bacteria directly enters into the brain and cause what? Meningitis. Second thing, the bacteria from there, from the nasopharynx and the oropharynx, it entered into your blood vessels, right, and caused meningococcemia. Meningococcemia. Not only that, once the bacteria is in the blood, you know this is called as bacteremia. Fourth important thing that was these endotoxins, they caused finally what? Fever, hypotension, all of you know, right? So they caused fever and they caused hypotension. So fever, hypotension, if they are too much, that condition you call it as shock. Fifth important thing was, in this patient, the fibrin content is so much increased that all the platelets will form a plug everywhere in the body, and that is called as disseminated intravascular coagulation. And sixth important thing, your platelets will be deficient, that is called as thrombocytopenia. So whenever the patient is having thrombocytopenia, so what will you look physically on the patient? You will see petechiae. You see classic petechiae on the surface of the skin. So what are the things, guys? What are the conditions that are associated? If I ask you what are the associated conditions with this bacteria? Associated conditions. So now all of you very perfectly know what I am talking. Associated conditions. Associated conditions are, yes, meningitis, meningococcemia, bacteremia, fever, hypotension, disseminated intravascular coagulation and thrombocytopenia. With the when patient is having meningitis, can anyone tell me what are the signs of meningitis? Signs of meningitis, you have to remember this, guys. Signs of meningitis are, one is nuchal rigidity. Patient will have nuchal rigidity. Next thing, patient will have positive Kernick sign. Positive Kernick sign. He will have positive Burzynski sign. I'm sorry if the spelling is wrong. Patient will have positive Kernick sign, positive Burzynski sign, right? Next, patient will have meningococcemia, bacteremia. So obviously when patient is having excess amount of bacteria in his body, patient will have sepsis, right? So all these things one has to definitely remember about this bacteria. The last and the very important thing I want to share with you, that this bacteria, guys, this bacteria, for example, this is your blood vessel, right? This is your blood vessel that is supplying your kidneys. This is the blood vessel that is supplying your kidneys, right? So when it supplies to your kidneys, on the surface of the kidneys, you have got a gland that is called as adrenal gland. Okay, so this gland is called as adrenal gland. So what is the thing? What is the thing is that in your body right now, there is excess amount of endotoxins, right? Patient is having meningococcemia, right? Excess amount of bacteria, excess amount of endotoxins. So all of them, for example, this is a renal artery. If this is a renal artery that is supplying to your kidney, all these endotoxins, they will go and damage your adrenal gland. Okay, they will cause infarction of the adrenal gland. So what will happen? What is happening is that the adrenal gland getting infarcted. This is called as adrenal infarction. So when your adrenal gland gets infarcted in the sense so severely that adrenal gland will blast like this. There is hemorrhage of adrenal gland. So when there is hemorrhage of adrenal gland, guys, what is happening is adrenal gland basically it releases some of the important hormones. For example, epinephrine, norepinephrine, aldosterone, cortisol. So these are some of the important hormones that are released by the adrenal gland. So right now when adrenal gland infarction has happened, hemorrhage has happened, do you think adrenal gland will release all this? No. So if adrenal gland is not releasing all this condition you call it as adrenal insufficiency. Sudden acute adrenal insufficiency. Suddenly all the adrenal hormones are decreased. Adrenal insufficiency. And this adrenal insufficiency is happening because of meningococcemia. So this condition is given a special name and this is called as Waterhouse Friedrich syndrome. Waterhouse Friedrich syndrome, right? So this is Waterhouse Friedrich syndrome or simply Waterhouse syndrome, because if you can't tell what is this, Friedrich, even I can't tell what is this, so fine, that is fine. It is Waterhouse syndrome, right? In Waterhouse syndrome, there will be acute adrenal insufficiency. And what is the etiology? Why? It is because of meningococcemia. It is because of meningococcemia, right? So what treatment, what is the diagnosis which you can do for this patient? How can you diagnose this patient? What is the diagnosis? How do I do the diagnosis is, the bacteria is in the CSF obviously because it entered into the meninges and caused meningitis, right? So what do I do is, I take a CSF sample from the blood and I do a gram staining. And when I do a gram staining, I will find out it is a gram negative bacteria. So this method is a gold standard. This is a gold standard method. Gold standard method. Okay. Next, when it comes to the treatment part, as simple as it. In treatment, I give three important drugs. One is called as ciprofloxacin. I give rifampicin. I give ceftriaxone. Ciprofloxacin, rifampicin, ceftriaxone are the three drugs which I give. So this is the end of the lecture, guys. Thank you so much.